Hello, our group will discuss about yogurt fermentation with mixed culture of Lactobacillus and Streptococcus. This is our group members, Elna, Eva, Mysie, Prisca, and Christhalia.
Yogurt is a fermented dairy product which is produced through the fermentation of milk by specific bacteria, mainly Lactobacillus dulbruetis subspecies bulgaricus and Streptococcus thermophilus. The fermentation process results in the production of lactic acid, which lowers the pH of milk, leading to protein coagulation and the development of yogurt's distinctive texture and tangy flavor. This experiment used microorganisms to investigate the yogurt making process, focusing on assessing the effect of varying milk and starter culture concentration on fermentation.
Now I'll explain the problem we addressed in our yogurt fermentation experiment, our research objectives, and a brief review of relevant literature. Yogurt fermentation enhances food quality and shelf life through microbial action. However, the best conditions like the right milk concentration and starter culture volume are not well defined. So, our study focuses on how these two factors affect yogurt's texture, acidity, smell, and color. Our objectives are first to demonstrate the use of microorganisms in food processing by using yogurt as a model system. Second, to explore the optimal methods and conditions for yogurt preparation. And third, to evaluate the effects of different milk and starter culture concentration to the yogurt fermentations. In the literature, Lactobacillus bulgaricus and Streptococcus thermophilus are shown to have a symbiotic relationship. Streptococcus thermophilus supports the growth of Lactobacillus bulgaricus, which helps produce amino acids and accelerate acidification. As fermentation lowers pH to around 4.6, casein coagulates to form the yogurt gel. Heating the milk before fermentation denatures proteins, which improves the final texture. Sensory attributes like flavor, aroma, and firmness are affected by milk composition and fermentation conditions. Lastly, the probiotics in yogurt contribute to digestion and help extend shelf life.
So now I, Elna Ellysia Epson with matrix number BS221100038 will explain about the methodology and results for our topic. So for methodology, firstly, a 500 ml of milk at 5%, 10% and 15% concentration was prepared. Then, the milk was heated slowly in a beaker up to 85 degrees Celsius and was maintained at that temperature for 2 minutes. The heated milk was cooled at ice bath until it reached 45 degrees Celsius. After that, a volume of 50 ml of each concentration was transferred into 2 sterilized 100 ml conical flasks in a laminar flow. The flasks were then immediately covered with aluminum foil. Then, a 0 ml, 2.5 ml, and 5 ml of starter culture, which is the yogurt, was added to each of the milk concentration in the flask containing cooled milk and was mixed evenly. Then the flasks were covered with aluminium and were sealed with parafilm. Then the flasks were labeled with the milk concentration, starter culture volume, replicate number, and date of the experiment. Then it was incubated at 42 degree overnight undisturbed. Observation was made the next day and recorded. So to obtain better and accurate result, we use two replicates of each milk concentration with the starter culture. So now I'll be talking about the result. In this experiment, the observation of the difference in yogurt after incubating overnight was obtained and tabulated in table 1 and 2. For every milk concentration, 2 replicates of yogurt with different volumes of starter culture, which were 0 ml, 2.5 ml, and 5 ml were aided and incubated overnight at 42 degrees Celsius, 3 to 6 hours undisturbed. The observations of before and after incubated overnight yogurt were observed and shown in Table 1. All of the yogurts show significant gel development, with the exception of the yogurts treated with 5% milk concentrations, which show only a little gel formation, and the yogurt with 0 ml of starting culture and 5% milk concentration, which shows no gel formation. Table 2 reveals yogurt's physical appearance, pH value, and scent. All yogurt replicates were found to be gel except for the control starter culture. The highest pH values were for the control starter culture, indicating less acid. The pH value increased with larger starter culture volume, reaching 4.09 to 4.20 when milk concentration increased. The yogurt had a sweet smell, creamy white color, and runny texture with the exception of the control starter culture at 5% milk concentration. The color of the yogurts was all creamy white with a hint of yellow, with the exception of the control starter culture with 5% milk concentration, which had the same color as before incubation. The texture of yogurt treated with starter culture at 5% milk concentration was curd and runny, while yogurt at 10% and 15% milk concentration was firm.
Hello, my name is Eva, and today I will be explaining about the first half of the discussion. So firstly, when comparing the fermentation outcomes across different milk concentrations, it's evident that higher milk content significantly improve yogurt quality. So for instance, based on the results, at 5%, the limited protein content hindered gel formation even with the presence of starter culture. In contrast, at 10% and 15%, gel formation was more consistent and firmer, indicating that increased protein availability enhanced the structural development of the yogurt. So the reduction in pH was most notable at 10%, showing efficient acid production by the starter cultures. However, the pH at 15% was slightly higher, likely due to the buffering capacity of the increased milk solids, despite ongoing fermentation. Aromatically, the yogurt evolved from a strong sour smell at 5% to a more pleasant sweet and sour aroma at 10%, and finally to a sweet or strongly sweet scent at 15%, highlighting improved fermentation performance. Interestingly, gel formation in the 15% control sample suggests the possibility of spontaneous fermentation by naturally occurring microbes. Based on the results in Table 1 and Table 2, these observations can be explained by the role of milk composition in yogurt fermentation. So for instance, at lower concentrations, the reduced protein content limits the ability to form a stable gel matrix, even when fermentation is initiated. So as the milk concentration increases, the presence of more solids supports both microbial growth and the development of a firmer texture. The drop in pH in the 10% samples reflects strong fermentation activity, while the slightly higher pH at 15% is attributed to the buffering effect of the increased solids, which resist sharp changes in acidity. Despite this, the fermentation remained effective, as shown by both the gel strength and the development of a sweet aroma. Next, the unexpected gel in the control sample at 15% highlights the possibility of contamination or natural fermentation due to lactic acid bacteria already present in the milk.
At 5% milk concentration, the yogurt was mostly curd and runny due to lower protein content, which led to weak gel formation. As we increase the milk concentration to 10% and 15%, the texture becomes noticeably firmer thanks to higher protein levels that supported stronger gel formation. In contrast, the control samples without culture remain runny across all concentrations, showing no signs of successful fermentation. Interestingly, most yogurt samples have sweet aroma while the control sample lost pure sourness as milk concentration increased. A few precautions to ensure successful yogurt fermentation. Firstly, it is important to use proper culture. We need to make sure it's within its expiry date and follow correct incubation guidelines. Next, clean sanitized equipment is essential to prevent contamination. In addition, good hygiene practices like wearing gloves and also lab coats help avoid cross-contamination. During processing, heat the milk to 85 Celsius to denature proteins and also reduce spoilage risk. Finally, incubate around 40 Celsius and monitor the pH. Once it's dropped to 4.6 or below, harmful bacteria are inhibited and safe fermentation is achieved. In conclusion, this research demonstrated how microorganisms, specifically lactic acid bacteria, play an important role in food processing through yogurt production. We observed that both milk concentration and starter culture amount significantly influence yogurt quality. Higher milk levels, combined with 2.5 to 5 ml of starter culture, resulted in firmer gels, better texture, lower pH, and also improved aroma. In contrast, low milk or no starchy culture lead to runny textures and higher pH levels. This highlights the importance of lactic acid bacteria in lowering pH and enabling milk protein coagulation during fermentation.